Guys, welcome on the heck in. We are talking over on the main screen. We're sciencing it up. We're talking about, this is not one of my good art pieces, by the way. We're talking about the immune response and how antibodies are generated to stain particular structures in the brain. So we're trying to get at how you can get, by the way, chat, follow Annabella. Go give Annabella that follow. Right now. You click that heart button right there. You click that heart button. You do the thing, chat. You do the thing. You do the thing. You get that, you hit that heart button. We're trying to dissect how you get these kinds of brain images from a microscope. How on earth do you do that, chat? And so we're talking about dissecting. So that's a whole mouse brain. We have stream avatars here too. Make sure, chat, you guys pick a stream avatar down there. The whales are very popular. The whales are very, very popular. So to stain the sample, first you have to make it transparent so you can see it totally see-through. And the reason we brought that up is because we actually have the microscope fired up tonight, guys. We are looking at, this is a sample of a butterfly antenna from a Monarch butterfly that we're looking at underneath the microscope. You can see the sensory bristles here. Those are the little hairs that are actually, they have neurons connected to them that run to the brain that tell the animal what it's smelling. Welcome on in, Shupluke. It's awesome. Do they do something like an MRI? So Shupluke, this is crazy time, okay? It's not even an MRI. They have this microscope called a light sheet microscope. Okay, let me show you guys what does a light sheet microscope even look like. And probably the best bet is for me to draw it out because it is hilariously complicated. Basically, what you end up doing is you make your sample see-through. This sample can be see-through by doing a process called active or passive clearing. And so what that means is all the protein gets ripped out of the cells and it becomes see-through. And then the microscope, your sample goes in overhead. And so there's this little vat here. And this vat has liquid in it. And then you have two cameras on either side of the sample. So you'll have here camera 1. And then you have a second camera here, cam 2. And then you submerge your sample into this liquid. So our sample, that was that brain. So you can just do a little brain like this. And what's hilarious enough, guys, is that you have to have a way to hook it.
Where's my
and
drink?
So it stays
What drink?
floating. Thank you for the hydrate, the cheese. I appreciate that. So it has to be like invisibility, Rip Chan. So what you do is you then add a connecting. You have to hook the sample so it stays floating. If you don't hook it correctly and it wiggles when you're trying to image it, it is going to be the biggest nightmare you can ever imagine, because it means your image will have these little jagged marks in it. I've tried doing this imaging on a sample that was... It was a very thick sample. What ended up happening was it was imaging for 22 hours. And at one point it jiggled and then was offset. So you couldn't actually match up the images anymore because of how offset that jiggle was. So some people hook it overhead. Other people will also do like a little underhook here. And then you start imaging your sample. And the way it images is it does the little slices. So every time it takes an image... Boop, boop, boop. That's one image, and then it does another slice, and then it does another slice, and it's doing the same thing from this other camera, from camera 2, slice, slice. It doesn't actually damage the sample. It's just going through different focal planes and imaging the sample bit by bit as it's going down the sample itself. And so you tell the software how thick of a sample you want to image, and the smaller the thickness, the better your images are going to be. That lets you get images like this, where there's the whole brain that you could be looking at. So that's on the right-hand side there. That's a mouse brain with neurons that are marked. Yes, like a scanner on a 3D printer. Exactly, Jen. It's the frustrating thing, though, what ends up happening is, again, like, the liquid that you submerge it, you have to have it be the same pH and like composition as either what the sample is or what you used to clear the sample. So passive clearing and active clearing are two liquids that you can put the sample in, and then you need to match the composition of that liquid with this liquid. It has to be the same composition, otherwise the optics get all screwy and it looks really, really bizarre. And then the question Big Gamey had was, well, how do we mark things? How do we make different like neurons light up? And it's actually a way, what we do is we hijack the immune response. There are tons of opportunities for error gen. The intro video that we have on the starting soon screen that has those ants that are 3D with like neurons in space, that's how these were imaged. Those ants were imaged. And those took, 13, 15 hours to do one sample. But what you do see is you get to see the neurons or whatever structure you're interested in 3D space and how it relates to other structures in the body. The other way of doing that is to put it on a confocal microscope, but that has a thickness limit. So here you're not limited by thickness per se, you're limited by how well you can clear the sample. If you don't clear the sample well, it's not going to work either, because with these cameras, there's also lasers here. So there's one laser here and one laser here that are shooting beams at the sample, and the lasers need to go through the sample all the way so both cameras can read the sample. And if it's not clear enough, what ends up happening is instead of the beam going all the way through, the beam will bounce, and if it bounces off, it's going to be totally distorted image-wise. So, and hilariously enough, chat raiders, there's not one good way of doing this. There are hundreds of techniques out there, and it seems like it's more specific to your organism question rather than it being different, like the same for different things. If that makes sense.
Where's my drink?
Thank you to Cheese for the hydrate.
What
Keeping
drink?
me hydrated, Cheese. Now, we were talking about, Big Gamey was wondering, how do we make some of those neurons light up? And so, I gave the example of, let's say we wanted to make some neurons light up in a fruit fly brain. And we were going to do this little goalpost structure here called the mushroom body. We'll just call it MB for short. Big Gamey, please interrupt me if you have any questions. That goes for anyone in chat. Because I need to know if y'all have questions so I can answer them. Our feelings on spiders. Rip Chance. I'm okay with spiders. We actually, Rip, if you stick around, I do have a spider preserved in amber that is 30 million years old. And so we can take a look at it under the microscope if you would like, Rip Chance. You just let me know, Rip. And we'll do it in just 1 second. Bit, ask all the questions. Everyone ask the questions. So, Big Gamey, when we were last, before the lovely raiders came in, we took these cells, the mushroom body cells, and we put them into the bunny rabbit. The bunny rabbit mounted an immune response against these cells because the bunny rabbit doesn't have fruit fly cells in it. It has bunny cells, so it had an immune response, and it generated these antibodies here. Now what we do, Big Gamey, is we harvest those antibodies. We bleed the rabbit. We get about 50 mils of blood, and then we have to undergo a couple of processing steps to clean up the blood, so it's just pure antibody. And you get the serum from that has the antibody cells in it. Then, Big Gamey, chat, you take your fruit fly brain, and you put your antibody juice on it. You do dilute it down. You don't have to have it as concentrated as you would have isolated your sample. You filter it down, and then you can dilute it. You can take 1 microliter of your antibody and 2,000 microliters of a balanced salt solution, right? So it's really, really, you dilute it down, and then you put that on your sample. So what happens, Big Gamey, is if everything worked well, your little antibodies are going to bind to your mushroom body. If everything worked well. And because if you prepped your sample correctly, if that's the only cell type you put into your bunny rabbit, then that's the only antigen that you'll have made. You will have made no other antigen in the body, right? Because you only injected mushroom body. If you start getting binding on other places, then your antibody wasn't very well made.
It's science.
It's science, Big Gamey. And then you'll need to start from scratch. But let's pretend that everything worked well, and that we're Gucci on that front, as Jern would say. We're totally Gucci. So to get it, this doesn't mean that it's glowing yet, Big Gaming.
Where's my drink?
Thank you, DeCheese, for the hydrate.
Drink?
This doesn't mean that it's glowing yet. In order to get it to glow, you have to make a secondary antibody, which means it's an antibody to recognize the antibody you put into your sample. And so we know that this blue antibody here is from rabbit. Oh. Rabbit. So your secondary antibody just needs to recognize rabbit pieces. And because there's no other rabbit inside the organism beyond this blue antibody, that's the only thing that your secondary antibody will bind to. So we'll make the secondary red. So it basically binds to your blue. Just like this. The rabbit, it's recognizing the rabbit antigen. And then what you do, Big Gamey, is you attach at the ends of these little the red bits, the secondary antibody, you have a fluorophore, which is a little something that lights up. I just made it green. It can be green. It can be, so green is GFP. You can have blue. You can have yellow. You can have red. So it is, let me see how well I can spell. Fluoro. Look at that. I may have written it correctly. That's what's attached here. It could be any color you really want. It has to be in the wavelength of light between 405 and 700 for most microscopes. And then basically what you do, you put the sample into the microscope. This laser here, well, now you want it to shoot the laser intensity that would excite the fluorophore that you put on. So remember that our fluorophore was green. That was attached to that antibody. So to get green to light up, you have to shoot a particular wavelength of light at it. So that's how I'm doing light here. And that light wavelength of light is 488 nanometers in length. You shoot the laser at that length, and then your sample lights up. Chat, Big Gamey, does that make any sense at all? I know that's like, we could totally have videos to also explain this, but I went the drawing route because there's a lot of complexities there and nuances that I'm trying to skip. But basically, you make something that recognizes your protein of interest, and then you make something that recognizes that thing that you made, and that thing that recognizes the next one has a little glow to it. What if you shoot too far? We'll slow that up. Ooh, if you shoot it too far, so if you do like, so reds are like 555 Big Gamey. You shoot it with that, it won't glow. But it's fine. You just change the laser. You just change the laser intensity, and then it'll glow. Normally, good fluorophores do not light up with other intensities of wavelength. There are some really, really bad ones out there, Big Gamey, that if you shoot like the laser intensity, it'll glow in different channels. So it's supposed to only glow green, but it also glows with red and blue. And that's okay if that's the only antibody that you put in. But let's say, Big Gamey, that there's, here's another structure. This is called the ellipsoid body. And let's say you want the ellipsoid body to also glow. And so you make antibodies to the ellipsoid body. And let's say you attach a...
In dreams, one is not
Good night, MB! Mike, thank you for the alert.
What does that mean?
Come.
You want that middle one to glow yellow, but then you need both to glow. I hope that makes sense. Guys, y'all want to look at a spider? Let's look at a spider. So, Big Gaming. Basically, you just need the ability... You need everything to be tightly lit up. Otherwise, it'll bleed through the two different things, the two different laser intensities, and then you can't tell them apart. And if you can't tell them apart, then you get a pretty picture, but you won't get information. Totally making sense. Oh, thank you, Schlupp. I appreciate you. My day job is a research scientist. I do stream. We do like interactive science streams here on Twitch 7 days a week.
Where's my drink?
Because I love science and I want to spread the science to more people.